Okay, we're almost to the end of our reproduction and development unit. We only have a couple more lessons. And today we're going to have a short lesson on cleavage and differentiation. So what happens to the zygote? Well, remember that the zygote is the fertilized egg. Well, we know that it undergoes mitosis and differentiation. But what does that mean, and where does that happen? So let's review where we are so far. We know through the process of meiosis, we get our gametes, which are our sperm cells with 23 chromosomes, and an egg with 23 chromosomes. The sperm and the egg meet in the fallopian tube, and that's where fertilization takes place. That forms a zygote, or a fertilized egg, with 46 chromosomes. And that's when cleavage takes place, through mitosis. So we go through several cell divisions until we have what's called a blastula, which is a hollow ball of identical cells, and that becomes a gastrula, which is where differentiation takes place. Layers form and cells become different from each other. This is differentiation. And we're going to talk about what that means, but all of our cells have 46 chromosomes, but we don't use all that information at the same time. We use some of that information for heart cells, some for skin cells, so on and so forth. And through differentiation, the embryo evolves and then continues to grow. So let's define some important vocabulary. Cleavage is the division of cells through the process of mitosis. And you should have learned this already or been introduced to it when we studied our vocabulary terms for this unit. So what happens is we have the zygote, which you know is the fertilized egg, and then it undergoes mitosis. And what that does is it produces two cells. And then those two cells undergo mitosis. And that produces 4 cells. And that undergoes mitosis. And that produces 8 cells. And that undergoes mitosis. And then that produces 16 cells. Forming a blastula, which is our next vocabulary term. So a blastula is a hollow ball of cells. That implants into the lining of the uterus. To begin development. Next, we have the gastrula, where identical cells start to position themselves into different layers. These layers are called germ layers. So here you see some cells move inward, producing a 3-layer embryo. So you have 1, 2, 3 layers, 1, 2, 3 layers. and then they migrate and they move around as they begin to differentiate. The formation of these layers is what helps mitosis or cleavage create cells that are the same but also different. Which brings us to differentiation. How can cells be the same but different at the same time? It almost sounds like an oxymoron. So we first have to know that all cells in the body have the same 46 chromosomes. But, and there's a big but, depending on where they are, they only use the genes with the information they need. For example, eye cells only use information from the eye genes. Skin cells use the information pertaining to the skin. But they both have the same 46 chromosomes. So that's what differentiation is. We all have the same genes. All our body cells have the same genes on the same 46 chromosomes, but depending on where they are in the body, it only uses the information that it needs. Okay, so make sure you can identify the following vocabulary in the diagram below. Well, there's a lot going on here, and we're going to cross these off as we use these. First of all, we know that the process of meiosis, so we're going to cross that out, produces both these things, gametes. Is that up here? Yep. And this is the egg, and this is the sperm. So gametes, it's these two right here, are sperm and egg. Then what happens is number 1 takes place. I'm going to label these. Number 1, fertilization takes place when the sperm penetrates the egg. So we got that. And then that forms a zygote, which has 46 chromosomes. Then 2, 3, 4, and 5. So 2 through 5 is cleavage. Well, which is mitosis. So each step. So this is mitosis number 1. This is mitosis number 2. Mitosis number 3. So every arrow represents mitosis. And this whole process is called cleavage, which results in what? A hollow ball of cells being formed, which is the blastula. So let's see, we used cleavage, blastula, mitosis. And then what's going to happen is this is going to implant into the uterine lining. And then what's taking place here, when you see the different layers, this is differentiation. They didn't make us label the gastrula. And I want to put a little star here. And then come down here. And please remember with differentiation, all cells have 46 chromosomes. Each, but they only use the information they need. Okay. And here you can see that the baby is developing. And then we have implantation, we have differentiation. So here, I'm going to make this a different color. I don't know if you can see this in here. Not letting me, there it is. I just made it like pink on mine. Okay, that is the placenta. So that's the placenta. And what happens is the diffusion of nutrients and waste. Diffusion of nutrients and waste across the placenta and then through the umbilical cord to the baby. So we got diffusion. It's moving from an area of high concentration to an area of low concentration. So the only one we didn't use was 23 chromosomes. And we're going to go back to here and put, I guess I'm going to try to put another little star next to the sperm and egg. And you can make a note down below, okay, sperm and egg have 23 chromosomes each. Okay, so the last thing that we're going to do is look at a couple of review questions that have to do with what we just talked about, which are on the next page. So this is a review of cleavage, which is the process of the fertilized zygote undergoing a series of mitotic divisions and reproducing the cells to form the blastula, the hollow ball of cells. So it says, although all of the cells of a human develop from one fertilized egg, the human is born with so many different types of cells. Which statement best explains this observation? 1. Developing cells may express different parts of their identical genetic instructions. 2. Mutations occur during development as a result of environmental conditions. 3. All cells have different genetic material, for some cells develop before other cells. Well, we know that all the genetic material is the same, so it's not that one. We don't know if all cells develop at the same time or not. We haven't talked about that. Mutations occur during the development as a result of environmental conditions, but that doesn't relate to this question. So the answer is 1. Developing cells may express different parts. Okay, that's the cells taking what they need of their identical, there it is, genetic instructions. So remember we said all 46 chromosomes are the same. Next question. Even though each body cell in an individual contains the same DNA, the functions of muscle cells and liver cells are not the same because 1. Mutations usually occur in genes when muscle cells divide. Lord, I hope not. Liver tissue develops before muscle tissue. We don't know that. Liver cells produce more oxygen than muscle cells. That has nothing to do with what we're talking about. Liver cells use different genes than muscle cells. There we go. That's the one we're looking for. And the last question. A human liver cell is very different in structure and function from a nerve cell in the same person. Why? Different genes function in each type of cell. Or liver cells can reproduce while the nerve cells cannot. We don't know that. Liver cells contain fewer chromosomes than nerve cells. No, we know that they all contain 46. Different DNA is present in each type of cell. No, we said all of them have the same DNA. So different genes function in each type of cell. So certain genes are turned on in liver cells and certain are turned on when they're in the nerve cell. Okay, that is our short lesson on cleavage and differentiation.